to be successful at developing new launch vehicles and ISS docking spacecraft, and for these capabilities to be pressed into service. As NASA works to procure services for logistics resupply of the ISS, planning should also include the needs of the ISS National Lab users. A productive ISS National Lab will require something like 20 to 25% of the transportation capacity, both up and down, with shipments spread across multiple flights each year. The ISS National Lab has enormous potential to advance the interests of the nation in commerce, science, medicine, technology, and education. If the steps I've just outlined are taken, I believe ISS National Lab will be productive and significant breakthroughs can be expected. Thank you for your time, and I look forward to answering any questions you may have.
Thank you, Dr. Stodek and Dr. Nickerson.
Inviting me to appear today before you to testify. My name is Cheryl Nickerson. I'm an associate professor in the Center for Infectious Diseases and Vaccinology at the Biodesign Institute at Arizona State University. I've been the principal investigator on multiple NASA-funded life science experiments. I serve as a consultant to the NASA Life Sciences Program at the Johnson Space Center, and I was honored to have been selected as a NASA astronaut candidate finalist in 2003. In your invitation letter today, you posed a series of questions to me regarding the utilization prospects of ISS research that I would like to now address in sequence. First, what have been the nature of my space-based research, and what have been my findings to date? My research focuses on understanding the molecular mechanisms of infectious disease, and specifically on the effect of spaceflight on the molecular mechanisms of infectious disease, with an important emphasis on the microbial pathogen response. Infectious disease is responsible for 35% of all deaths globally, and it's the world's largest killer of children and young adults. The economic impact on the U.S. alone from infectious disease exceeds $120 billion annually. And of course, future threats loom on the horizon for us globally as well, including new and re-emerging infectious diseases for which we do not have sufficient treatments, antibiotic-resistant strains, and always the potential for the intentional misuse of microbial pathogens as agents of bioterrorism. Clearly then, new treatment and prevention paradigms are desperately needed. My spaceflight research is focused on the bacterial pathogen Salmonella, which is a global threat to public health and accounts for approximately 30% of all deaths from foodborne illness in the United States. From NASA's perspective, Salmonella is considered a potential source of infection during spaceflight that could incapacitate crew members during a mission. However, there are currently no human vaccines to prevent Salmonella foodborne illness. I applaud NASA's foresight in funding our spaceflight research in the field of infectious diseases, as well as the awesome engineers for building this wonderful ISS craft. The connection between spaceflight and infectious diseases was not immediately clear 10 years ago when NASA originally funded our research. Based on our early findings using ground-based spaceflight analogs, NASA awarded us a grant to investigate the effect of true spaceflight on Salmonella disease-causing ability, which we call virulence, and gene expression results. This experiment flew on STS-115 in September of '06, and the results were remarkable. They showed that spaceflight increased the virulence of this important human pathogen and globally altered its gene expression profiles by changing 167 genes. Now, the interesting part about that is it not only increased its disease-causing potential and changed all these expression of genes, but it did so in unique ways that are not observed using traditional experimental approaches in the laboratory. So what we've done is we've been able to discover new ways that this important human pathogen causes disease in the host. We also discovered a key master regulatory mechanism that controls the vast majority of Salmonella's responses to the spaceflight environment. This molecular target, along with others that we identified in my laboratory, holds real potential to be translated into new therapeutics and new vaccines to treat and prevent human enteric foodborne illness caused by Salmonella. Our findings were published in the Proceedings of the National Academy of Sciences. The success of our flight experiment on STS-115 inspired a follow-up experiment on STS-123, which just flew in March 2008, and the results from that are equally exciting, and we're looking forward to publishing those within the next 1 to 2 months. An important part of our spaceflight work is helping us to understand, therefore, how microbial pathogens cause infectious disease here on Earth. And likewise, then using spaceflight as a novel enabling research platform to translate those innovations into infectious disease control here for the general public on Earth. Shortly, we expect NASA and the public to receive a direct benefit from their investment in our work. Second, what is my perspective on the future potential for the use of microgravity environment as a research tool? It's simple. The microgravity of spaceflight offers a unique environment for groundbreaking biotechnology and biomedical advances and discoveries to globally advance human health, not just in infectious disease, but in some of the major health illnesses here that we worry about globally. Cancer, aging, bone and muscle wasting diseases, and tissue engineering. And it will have a long-lasting impact on our nation's scientific capability, economy, and the quality of our lives. Many breakthroughs in life sciences research have come from studying living systems in extreme environments. The environment of spaceflight offers insight into fundamental cellular and molecular response mechanisms that are directly relevant to human health and disease and which cannot be observed using traditional experimental approaches in the lab. Third, and my final question, what are any potential applications of the basic research I've conducted to date or intend to pursue? The investment that NASA has made in our research for innovations in infectious disease treatment and control will provide long-lasting return in the protection of humans as they explore space and for the general public here on Earth. An example of a potential boon from spaceflight experiments is my laboratory's discovery that gene regulatory proteins participate in the spaceflight response of microbial pathogens. Gene regulatory proteins affect every property of a cell, including its ability to cause disease. My lab is currently studying how this regulatory and these regulatory pathways work in salmonella and other important human pathogens and how they can be manipulated to control microbial virulence and subsequently how those findings can be translated to the design of new drugs and new vaccines with clinical applications. One key to our nation's economic success has been its ability to consistently provide unique answers to the world's problems. We have the opportunity here and now to advance in a field where the U.S. is a world leader. I firmly believe that space exploration and development will be one of the defining activities of our nation that will lead the world in this new millennium. And I thank you.
Thank you, Dr. Nickerson. Mr. Pickens.
Mr. Chairman and members of the subcommittee, thank you for the opportunity to make my first appearance before you today as the Chairman and CEO of Space Hab to discuss the significance of NASA's International Space Station program, not just as it applies to commercial aerospace company, but also for its tremendous potential to benefit mankind. I represent the commercial perspective of microgravity and its value. First question was, how do your company arrive at the decision to pursue vaccine development under microgravity conditions